to you all. In today's class, we are going to see the chapter 2, part 1, biological classification. So, last class we studied about the unit 1, chapter 1, that is 7 kingdom classifications. So, we are having a variety of new names, that is the genus and species. It will be very new to you. So, go through it, write and see it very often. Okay. So now let's get into today's session. When we talk about the biological classification, we have to look over the historical background, how the classification system was evolved, and which is the universal one we are following right now. Okay, so why we need a classification? Coming to the living organisms, apart from the humans, plants and animals, we are having millions and trillions of microbes around us, that is the microorganisms, are also playing an important role in the universe. Is it right? So these fungus, bacteria, everything, virus has to be placed in a particular order so that it will be easy for the scientists to do further researches. Okay, so the classification, how they are dividing, means based upon the eras, based upon the scientific discoveries, we are dividing the classifications into 2 kingdom, 3 kingdom, 4 kingdom, 5 kingdom, even 6, 7 and 8 kingdom classifications are also there. Okay, so after the discovery of microscope, some classification arises. So before that, some classification was found. So how the classifications was evolved. Okay, so that is what we are going to see in the chapter. So now we are going to see the classifications one by one. The earliest classification, that is in the even when the start of civilization itself, the scientist Aristotle, so he classified the living organisms into two types. One is plant and another one is animals. Okay, so in plants he included unicellular, multicellular organisms. Further, he classified the tall plants as trees, as well as small as shrubs, and very small plant as herbs. Okay, so this is the basic classification. Based upon the morphology, he classified the plants as tall, short and very short, that is trees, shrubs and herbs. Okay, so then he classified the animals as red blood animals and others without having a red blood, and also he classified based upon the living places. So the some birds can live in the air, some can live in the land, some can live in the water, etc. So based upon that, he gave this classification. This is the very basic classification. Okay, so the next game, our scientist, that is the taxonomic scientist Linnaeus, so he further classified the system again into two types. One is said to be plant and animals. Okay, what Aristotle did based upon the morphology, he classified. Is it right? So here the scientist Linnaeus, he included some other characters and he classified plants and animals. That is, he studied about the nutrition and locomotion. Okay, so first one is morphology alone. In second classification, he included the study of nutrition and locomotion. Okay, so he classified it into two types, plants and animals, based upon the locomotion as well as the nutrition. Okay, so he placed unicellular protozoans and multicellular protozoans into animals groups. Okay, so because of the locomotion, okay, plants which cannot able to mobile, which cannot able to move, is it right? So he separated the plants into a separate group. Okay, clear. So but this gave confusion to many scientists because he left so many organisms. For example, if you take the euglena, okay, so which is having both the properties of plants as well as animals. For example, this euglena, which is having a process, that is a photosynthesis, it can able to perform the process of photosynthesis, but there is an absence of cell wall, same like the animals. Okay, so but he didn't place euglena anywhere. And also the fungi, okay, so they take nourishment from the dead and decaying organisms, and it was not placed. So he placed all bacteria, blue green algae, fungi, everything under the plants. If you see here, you can able to identify the bacteria, fungi, everything under the plant categories he kept. But under the animal categories, he kept algae. So there is no proper classifications. Okay, this was not adequate to keep all the living organisms. So then came the three kingdom classification which was put forward by the scientist Ernest Haeckel. Okay, so he is the German investigator Ernest Haeckel. So he proposed the three kingdom classification. He included one more group or one more kingdom. So here these we are saying it as kingdom. Okay, so Animalia, Plantae, Protista, everything we have to give the name kingdom Animalia, kingdom Plantae, kingdom Protista. Okay, so he included one more group that is said to be the Protista. Here for the first time he introduced protozoa, fungi, bacteria and all the other microorganisms under the group Protista. Still the fungi was placed in the same group. There is no separate group for the fungi because we are having a vast number of properties which was differentiating bacteria as well as the higher organisms and fungi. Okay, so the next classification, that is the four kingdom classification, was given by Copeland. So he proposed one more group, that is he gave Protista, MONERA, Metaphyta, Metazoa. Metaphyta and Metazoa is nothing but a plant and animalia. Okay, so he included the group that is said to be the MONERA. Okay, so in this MONERA he differentiated for the first time prokaryotes and eukaryotes. So this MONERA included the prokaryotes and the Protista, which includes the unicellular eukaryotes. I already told in the first class the prokaryotes are always unicellular. Okay, so that will come under MONERA. But eukaryotes can be either unicellular or else multicellular. Okay, so some will come under Protista and some will come under the MONERA group. So in this two kingdom classifications proposed by Aristotle and Linnaeus. Okay, and also the three kingdom classification by Ernest Haeckel. So every classifications, 2, 3 and 4 classifications, there is no proper group for the fungi. Okay, so everything was demolished. So after the discovery of the microscope and the invention in cell biology, and the scientists, they studied the cell in depth after the discovery of the microscope. Okay, so later in the year 1969, so our Whittaker, he gave us the five kingdom classifications. Okay, so it is the first classification which was universally accepted. Okay, till in this 20th century also we are following this classification. Advances in cell biology and evolutionary biology lead to the scientists to put forward the questions how these two kingdom classification, the three kingdom classifications worked out, and what are all the drawbacks. Okay, so in the year 1969 this Whittaker proposed the five kingdom classification. So he classified the organisms based upon the five properties. Let us see one by one. So first one is complexity of the cell. Okay, so complexity of the cell, which includes on the basis of their nucleus and other characteristics, there are two divisions are there. One is said to be a prokaryote and another one is said to be an eukaryote. So okay, so in this MONERA we are having a prokaryotic organism. So based upon the cell structure. Okay, the next one is based upon the complexity of the organism. Complexity of the organisms means earliest were unicellular organism. Okay, unicellular, we are having a single cell, that is said to be the unicellular, and eukaryotes were evolved. Okay, we know that the first formed living organism is an unicellular, and due to the evolution only our eukaryotes is getting evolved. Is it right? So that eukaryotes evolved into a multicellular organism. First one is unicellular, then eukaryotes, and eukaryotes to multicellular organisms. So in this complexity of organisms, that is we are saying it as a Protista. Lowest group is MONERA, which include prokaryotic unicellular organisms, and coming to the Protista, which includes eukaryotic unicellular as well as the multicellular organisms. Okay, the next, based upon the mode and source of nutrition as well as the ecological role, that is the based upon the biological role, we are classifying the organisms into the kingdoms into three types. We know that plantae, animalia, as well as the fungi. Okay, so here coming to the mode of nutrition, we know that we are having two mode of nutritions. One is said to be autotrophs and another one is said to be the heterotrophs. Okay, so here you see the autotrophs. Autotrophs means, listen carefully, autotrophs means the organism which can able to synthesize their own food material. Okay, that is said to be the autotrophs. For example, you take plants, they can able to synthesize their own food material with the help of the sunlight, is it right? So plants will come under autotrophs. If you are taking the heterotrophic organisms, animals and fungi will come under the heterotrophs. For example, heterotrophs means the organisms which depends on the other organisms. For example, if you take animals, it depends on obviously on plants or some other dead and decayed matter. This is right. If you take a fungi, so which will depends on the dead and decaying matters only. So animals as well as the fungi will come under the heterotrophs. So even we can also divide this heterotrophs into some other types also. So we might have studied in the lower classes. One is said to be holozoic, is it right? So what is a holozoic? Holozoic means it intake the solid food materials and breaks into simpler form. Okay, so some fungi and as well as some animals will come under the holozoic categories. So it will intake the solid food materials and it will further break down. Okay, so and most we have studied this one, saprophytic, is it right? So what is saprophytic? Saprophytic organisms which will obtain their food materials from the dead and decaying organic matter. Okay, so that also come under heterozoites, and another one is parasites. Okay, so parasites means which also we have studied in our lower classes. So here the parasitic nutrition, how this organism taking the nutrition means it will partial or completely on the other organisms for their food, is it right? So these are all the categories of heterotrophs. So based upon the mode and the source of nutrition, that is whether the organism is autotroph. If the organism is autotroph, they can able to produce the food, hence we are saying it as a producer. Okay, heterotrophs, it will consume the producer, that is we are saying it as a consumer. And decomposer, decomposers means it will decompose the dead and decayed organisms. As I told, saprophytic, parasites, everything will come under the decomposers. So decomposers plays a vital role in the maintenance of ecology. Okay, so based upon these five categories only, that is the complexity of the cell as well as the complexity of the organism and mode and source of nutrition. Okay, so he put forward this group. I will explain once again the producers. The plant which utilizes the light energy, water, okay, to synthesize this organic food, that is said to be the producer. And consumers, we are saying the animals that consume, it will either consume a plant or a lower animals to survive, is it right? So decomposers, coming to the decomposers, fungi or some bacteria which also depends on the plants and animals for their food and decompose them, that is said to be the decomposers. Okay, so now we are going to see the groups one by one. First group, we are in this video session, we are going to see only about the MONERA, kingdom MONERA. Okay, so it was the first formed organism, as I told earlier, MONERA, which includes the prokaryotic organism. They are always unicellular in nature. Okay, so it is the earliest group. Okay, so they are microscopic, like the bacteria which we can see only under the microscope, is it right? If they are found in all over the earth, okay, they are said to be omnipresent. Okay, even in the moist environment, even in the deep sea as well as in the hot springs, these organisms were found. If you see, this is the structure of this bacteria. Okay, I have given two diagrams here. One is said to be the bacteria and another one is said to be the eukaryotes. Okay, so now coming to this bacteria, here we don't have any definite nucleus. Hence why we are saying it as a prokaryotes. Easily you can remember P and B for bacteria. If prokaryotes means it includes bacteria, so which does not have any proper cell wall or we can say there is no definite nucleus, okay, but the nuclear material, the DNA, which was scattered in the cytoplasm, there is no nuclear membrane, etc. So hence why it was scattered freely in the cytoplasm. If you see the eukaryotic cell, we are having a proper nucleus, and inside the nucleus the DNA was present, okay. So here in this prokaryotes, we are having a no bound organelles was not present in the prokaryotes, but in case of eukaryotes we are having a well bound organelles, okay. And also the cell wall, here the bacterial cell wall we are having the peptidoglycan layer, but in case of eukaryotes we will be having a polysaccharide layer, chitin will be there, so some other molecules will be present. But in case of bacteria, most of the bacteria are having a peptidoglycan layer, only some virulent bacteria is having the polysaccharide layer, okay. So based upon the layers present, that is the cell walls, we are classifying the bacteria into two types. One is said to be gram positive and another one is said to be gram negative bacteria. So this was discovered by the scientist Christian Gram, Hans Christian Gram. Hence why we are giving the name as gram positive and gram negative. We know that the bacteria which is not having any definite cell wall, we are saying it as a cell layer. So in this cell layer, so based upon the peptidoglycan layer present in the bacteria, we are dividing it into two, okay. So if you take the gram positive bacteria, we are having a very thick peptidoglycan layer, that is the outer membrane. But coming to the gram negative bacteria, this violet color indication is said to be the peptidoglycan layer, okay. So here we are having a very less peptidoglycan layer. So this is one classification of bacteria. So under kingdom MONERA we are seeing a bacteria as an example. I have told bacteria is the first formed living organism. It is a unicellular, it is a prokaryote, it is present everywhere, even in the hot thermal vents, even in volcanoes it was present, even in the sea it was present. It was present in my hand, it was present in your face, okay. So everywhere this bacteria was spread across, okay. I have given two diagrams for your understanding for unicellular, that is the prokaryotes and eukaryotes. Prokaryotes, coming to the prokaryotes, we are not having any well developed nucleus and well developed membrane bound organelles compared to eukaryotes, and the cell wall is also absent, only the cell layers are present in order to protect the internal organelles, okay. So some bacteria we are having a flagellum and some bacteria we are having a phylum, okay. So this flagella is used for the movement and this phylum is used for the reproduction, okay. So next, based upon the peptidoglycan layer, we are dividing the bacteria into gram positive as well as the gram negative. Thick peptidoglycan layer means it is said to be gram positive, and thin layer means it is said to be the gram negative bacteria. And also coming to the shape, based upon the shape we are classifying the bacteria into different types. We will see one by one. The first one is said to be the cocci, okay. In singular we are saying it as a cocci, but in plural we have to say as coccus. The same for bacilli also. If the bacteria is spherical in shape, then that is said to be the coccus. For example, you can take a staphylococcus. It will be like a grapes. If you see the structure, it will be like a grapes because it is spherical in shape. If you see under the microscope, it will be a spherical shape. That is, we are saying it as a cocci. Okay, cocci shape. Okay. The next one is a rod-shaped bacteria. That is said to be the bacilli, okay. We are saying as bacillus, okay. For if you take an example, streptobacillus, diptobacillus, these are all the examples of rod shaped bacteria. We are saying it as a bacillus, okay. The next one is curved shaped bacteria, that is a comma shaped bacteria. If you take Vibrio cholerae, which is an example for cholera causing bacteria, so that will be in comma shape. We are saying it is comma shaped bacteria, that is Vibrio cholerae, vibrous. The next one is spiral shaped bacteria. We are saying it as a spirochetes, okay. They are having their shapes will be like a spiral, okay. It will be like a coiled shape, that is said to be the spiral, okay. The next one is we are seeing the reproduction. So how the reproduction will happen? We know that the reproduction of the bacteria mostly happen by means of asexual means, and some by means of the sexual means. Okay, so if you see the asexual reproduction, so first we will see this one, asexual reproduction. We know that the binary fission will takes place, in which the DNA will duplicate, and the each daughter cells, here the DNA is there, it is getting duplicated, and each daughter cell will receive the DNA from each daughter cells receives each one. Okay, is it right? So thus hence they are genetically similar. Okay, there is no genetic variation because it is dividing as such. But when you come to the sexual reproductions, by means of conjugation this process will take place. Is that right? The bacteria, the two bacteria will come and contact with each other by means of this pili, and the DNA material will get transferred from one bacteria to another bacteria. Okay, so these are all the two modes of reproductions. So here coming to the reproduction, we have studied two types. One is asexual and another one is sexual. Asexual by means of binary and sexual by means of conjugation. Is it right? So this MONERA, how they will intake the food means, I have told some are autotrophic and some more heterotrophic. Okay. So now we are classifying this kingdom MONERA basically into two sub classifications. One is said to be Archaebacteria and another one is said to be Eubacteria. The Archaebacteria, which includes the anaerobes. Okay, anaerobes means in the absence of oxygen only these organisms can able to survive. Okay, so in the absence of oxygen means, imagine, so under the sea also it can able to live, in the volcanoes also it can able to live. Okay, so we will see one by one. Thermoacidophils, halotolerant and methanogens. Okay, phils, phile means loving. Okay, so which can able to live in the condition. Thermo means high heat. Okay, acidic condition. So even in the presence of high temperature as well as the high acidic nature, the organisms can able to live. Okay, that is said to be thermoacidophils. If you see this picture, this is the volcano. Even in the volcano, which is having the temperature of 2500 degrees Celsius, it can able to survive. Okay, so such bacteria are having a special type of cell walls, hence they can able to even survive in the hot winds as well as in the sea also. Halotolerant organisms under the sea also, some microorganisms, that is some bacteria are surviving, that is to be the Archaebacteria. Halo means salt. Okay, it can able to withstand the salt and it can able to live. Next one is methanogens. Methanogens are an anaerobes which lives in the guts of the cattle like cows and buffaloes. Okay, they are used for the biogas productions. The methanogens from the dungs of these cattle are used as a source for biogas. Okay, so in these all thermoacidophils, halotolerant and methanogens, the bacteria cell wall is little different when compared to other, hence why it can able to live in the extreme hard condition. As I have told, it is the first formed living organism. Due to the evolution, that archaebacterial cell wall was getting lightly changed because of the external factors, and we are saying nowadays bacteria as an eubacteria. Okay, that is the true bacteria which is having a cell wall as well as the flagella. So coming to this eubacteria, we are having an example of cyanobacteria. Okay, first we will see the cyanobacteria. A cyanobacteria is otherwise called as a blue-green algae. Okay, so here this cyanobacteria which contain a chlorophyll and other pigments. Okay, so under aquatic in nature, they were a part of a kingdom Plantae. But earlier, because they are prokaryotes, they were placed in the kingdom MONERA. Okay, so they are said to be photosynthetic autotrophs. As I have told, we are having an autotrophs as well as the heterotrophs. Is this right? The autotrophs are further classified into photosynthetic as well as the chemosynthetic autotrophs. Photosynthetic autotrophs means they will utilize the sunlight. Okay, and they can able to synthesize their ATP molecules. Chemosynthetic autotrophs means they are particular organisms which will utilize the inorganic substances like nitrates, chlorides. So these are all the inorganic substances and they will utilize it, and they will produce the ATP molecules. Okay, so these cyanobacteria which can able to live in the freshwater, marine as well as the terrestrial organisms. They are said to be unicellular as well as the filamentous organism. Okay, so if you see the colonies of blue-green algae, if you are growing the bacteria in an artificial medium, means you will be getting a colonies, etc. So these colonies are surrounded by a gelatinous sheath membrane. Okay, so some of this Eubacteria, that is the cyanobacteria, are used for a nitrogen fixation process, okay, which was helpful in the agriculture also. So they are used for nitrogen fixation. That is example is Nasdaq, Annabella. We can say some other examples also. This heterotrophic bacteria, I have told that most of the bacteria are decomposers. Okay, so even though these bacteria plays an important role in decomposition process, some bacteria are helpful for human also. Okay, we are using some bacteria in our day-to-day life also. For example, if you take the curd, okay, that is for making the curd we are using lactobacillus, but we don't know whether that is a lactobacillus or not. If you take a, see here we are having a inoculum which contains a curd. If you are inoculating it into the milk, after 8 hours the fermentation process will take place and our milk will curdle into curd, etc. So that for this reason we are using the lactobacillus. This spoonful of curd contains only bacteria which is lactobacillus. So this is very useful for our day-to-day life, for our cheese making, butter, for everything we are using only the bacteria. Okay, and also antibiotic production. So for if you are going for any bacterial infection to the hospital, means the doctor will prescribe the antibiotics, etc. So that antibiotics was prepared only by means of, with the help of bacteria only. Next one is for fixing the nitrogen. I have told earlier this cyanobacteria, the nostoc, Annabella, all these living organisms will fix the atmospheric nitrogen to the root of the plants and helpful in growing the plant. Okay, so these are all the applications. Coming to this mycoplasma, mycoplasma is the smallest living organism which is not having any cell wall. Okay, so it is a pathogenic organism. Mostly it will cause disease to plants and animals. Okay, so next, if you see the in your book page number 17, you will be having on tablet column, that is the characteristics of five kingdom classifications. So today we have studied about MONERA, is it right? So I will explain only about this MONERA today. Later we will see it in the later classes. Okay, if you see the cell type, we know that it is a prokaryotic organism. I have told the cell wall which is made up of a polysaccharide layer, is it right? So based upon the polysaccharide layer, that is we are saying it is a peptidoglycan. Okay, that is come under polysaccharide. If you see the nuclear membrane, absent of nuclear membrane, and body organization is very cellular, and mode of nutrition autotrophic. Some are chemosynthetic, that is the chemoautotrophic, and some are photosynthetic autotrophs, is it right? And some are heterotrophic organisms. So this is the tablet column for MONERA. So next class we will see about the protista fungi and we will see the tablet column of this one. Okay, so go through the book once or twice. Okay.